Hello everyone. Today we discuss staining techniques in microbiology, a core laboratory concept that helps us visualize tiny microorganisms under the microscope. Microorganisms like bacteria are extremely small and often hard to see. Staining lets us make them visible and study their structures in detail. Staining enhances contrast in microscopic images. Without stain, many bacteria appear transparent because their refractive index closely matches water. In this video, we'll cover what staining is, why it matters, key terminology, and the main staining methods used in labs and clinics. We will highlight gram, acid-fast, capsule, and endospore staining, explaining steps, interpretation, and clinical relevance for each method. Throughout, you'll see clear microscope images, labeled diagrams, and concise step summaries to help you apply these techniques safely and accurately. First, a stain is a dye that binds to cellular components to increase visibility under a microscope. A simple stain uses one dye to reveal shape and arrangement. It does not differentiate between bacterial types. Differential staining uses multiple reagents to separate organisms into groups based on cell wall or structure differences. Key reagents include a mordant that fixes dye, a decolorizer that removes stain selectively, and a counterstain for contrast. Fixation stabilizes cells on the slide, preventing loss or distortion during staining and improving dye uptake. Understanding these terms helps interpret results correctly and choose the right protocol for diagnostic or research purposes. Let's examine 4 essential staining methods: gram staining, acid-fast, capsule, and endospore staining, and when to use each. Gram staining differentiates bacteria by cell wall structure. Results are gram-positive purple or gram-negative pink after counterstain. A typical gram sequence is crystal violet, iodine mordant, alcohol decolorizer, then safranin counterstain for clear differentiation. Acid-fast staining detects mycobacteria with waxy cell walls. Primary stain and heat or detergent help penetrate the cell wall before decolorization. Capsule staining highlights polysaccharide capsules that resist staining. Negative stains or special stains create a clear halo around stained cells. Endospore staining identifies resistant spores inside bacteria. Spores take up primary stain while vegetative cells are counterstained for contrast. Gram results guide initial antibiotic choices and infection control. For example, gram-positive cocci suggests staphylococci or streptococci. Acid-fast positive results can indicate tuberculosis or atypical mycobacterial infections and trigger specific public health and treatment actions. Capsule detection is important for organisms like Klebsiella and Cryptococcus, where capsule presence links to virulence and treatment decisions. Endospore identification helps detect spore-forming pathogens such as Bacillus and Clostridium, which require special handling and clinical awareness. Remember limitations. Staining gives preliminary data, but often needs culture, molecular tests, or susceptibility testing to confirm diagnosis. Good laboratory practice, reagent quality, and trained personnel are essential to ensure accurate staining and reliable clinical interpretation. Quick summary: staining improves contrast, reveals structures, and differentiates organisms to support diagnosis and research. Practical tips: always fix slides properly, follow reagent timings, use positive and negative controls, and document procedures for reproducibility. Safety note: handle dyes and fixatives with care, wear PPE, and dispose of biological and chemical waste according to laboratory regulations. Useful workflow: prepare sample, fix, stain, rinse, dry, and examine under oil immersion when high magnification is required. If results are unclear, repeat with controls, verify reagent integrity, or apply complementary methods such as culture or PCR to confirm findings. Thank you for watching. Use these staining principles to enhance your microscopy practice and consult your laboratory manual for specific protocols. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, kinetic, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.